Hello, everyone, and welcome to today's webinar, Advances and Challenges in Targeting Kinases in Cancer Therapeutics. I am Aishath of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BPS Bioscience. To learn more, visit bpsbioscience.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Aishath Naeem, postdoctoral fellow, Dana Farber Institute at Harvard. Aishath, you may now begin your presentation.
Thank you, and thank you for the invitation. So my talk today is about targeting BTK in CLL. And I'll start with a brief introduction on chronic lymphocytic leukemia, or CLL. So what is CLL? CLL is the most common B lymphoid malignancy in North America. In 2022, new CLL cases is estimated to be at 20,160, which represent about 1.1% of all new cancer cases in the United States. The median age of diagnosis is 70 years, with only a small fraction of patients younger than 45 years. CLL is an indolent or slow-growing malignancy characterized by clonal proliferation and accumulation of mature B cells within the blood, bone marrow, lymph nodes, and spleen. This is a very slow-growing disease. People can usually sort of have normal life with CLL for a very long time, be asymptomatic before the disease can pick up momentum, and there is a need for medical intervention. So treatment is often initiated only in the presence of active symptomatic disease. So let's talk about risk factors and driver events in CLL. 80% of the CLL patients exhibit one of the 4 chromosomal abnormalities. There are deletions on 11q, 13q, 17p, and trisomy 12. So these events can be biological predictors to patients' response to therapy. Another important predictor is the IGHV mutation status. So there are 2 subtypes of CLL, and these can be seen in the schematic on the slide. The subtype that's unmutated with minimal or no hypermutation in the antibody gene, and the subtype that's mutated with much more somatic hypermutation. So the IGHV mutational status discriminates between the more mature, genetically stable CLL and the more immature, genetically unstable CLL. So patients with unmutated IGHV have a more aggressive disease course than those with mutated IGHV. So we have over 200 candidate genetic drivers identified so far. With increasing amounts of CLL being sequenced, we are learning more about the driver events and their impact on disease relapse and the clinical outcome. So this just shows a snapshot of the evolution of the CLL treatments over time. And as you can see, the therapeutic landscape of CLL has significantly evolved as our understanding of this disease has advanced. The current CLL treatment includes chemotherapy agents, inhibitors of the BCR pathway, and an inhibitor that targets cell death pathway. Now, targeted therapy, which has picked up in the last decade, is a cornerstone of precision medicine. It's really transforming CLL treatment by focusing on specific molecular or drivers of the disease. And these treatments have significantly improved the survival rates of CLL patients. The BCR signaling pathway. So the B cell receptor, a component of the pathway, is important for the proliferation, maturation, and survival of normal B cells. So how does this pathway get activated? On antigen binding to BCR, CD79A and B, which are non-covalently associated with the receptor, gets phosphorylated by Syk and Lin, which leads to formation of a complex that involves BTK. Now, BTK tyrosine kinase is a member of the TEC family kinases and plays an essential role in B lymphocyte development and function. And as you will see, BTK is a critical mediator in this pathway. So as I mentioned before, after antigen engagement, Syk and Lin phosphorylate CD79A and B, which phosphorylates BTK at the tyrosine 551 site. And then BTK gets autophosphorylated at tyrosine 223. So now the immediate downstream substrate of BTK is PLC gamma 2, which then gets phosphorylated by BTK. And this triggers a cascade of events, all signaling down from the BCR pathway. And this pathway activates transcription factors that are significant for decisions such as proliferation, survival, apoptosis, and migration of B cells. So BTK enhances the signal between the B cell receptor and the nucleus. And in CLL, BTK is overexpressed both at transcript and protein level compared to normal B cells, leading to an amplification of the BCR signaling pathway. An amplification of this pathway leads to increased pro-survival signals through its effects on PA3 kinase, PLC gamma 2, and NF-kappa B. And this leads to these survival pathways are essential for the survival of the CLL cells. So this led to the development of specific inhibitors that target BTK. The first group of BTK inhibitors are covalent inhibitors. They're called covalent because they bind to their target by forming a covalent bond. Ibrutinib was the first of his class, the first one that came out. And while the efficacy of this drug is very good, long-term treatment can be associated with toxicities, and in some cases, these toxicities can be quite severe. We also have the next generation inhibitors in this same class, acalabrutinib and zanubrutinib. So here we can see the kinome selectivity among the covalent inhibitors. So the size of the red circle is proportional to the degree of inhibition. And as you can see, ibrutinib has activity against quite a few kinases, which may explain some of the toxicities observed with this drug. Zanubrutinib has activity against much fewer kinases than ibrutinib, but acalabrutinib is the most specific of the covalent inhibitors. And all three have the same binding mechanism. As shown in the diagram below, they all bind to the C481 site found within the ATP binding pocket in the kinase domain of BTK. And another problem that arises with this group of inhibitors, with ibrutinib specifically, is despite clinical success, long-term treatment can lead to acquired resistance, with the most commonly reported one being the C481S mutations. And we also have the less frequently identified activating mutations in the downstream substrate of BTK, which is PLC gamma 2. So presence of this mutation at this site abrogates the covalent binding of all of these drugs. So this led to the development of non-covalent BTK inhibitors. These were designed to bind to BTK without relying on the C481 site, thus providing options for those patients that has progressed with the BTK C481S mutations. So we have a few in preclinical development, and two of these, pirtobrutinib and nemtabrutinib, are currently in clinical trials. And here you can see the kinome profile of the two non-covalent inhibitors. And as shown in the graphs, pirtobrutinib is highly more selective for BTK compared to nemtabrutinib, with 300-fold selectivity for BTK versus 370 other kinases. So this is a timeline of the BTK inhibitors so far. So as I mentioned before, ibrutinib was the first of its class to come out. In 2014, it was FDA approved for patients with CLL who have received at least one prior line of therapy. In 2016, it was approved as an initial treatment. In 2019, acalabrutinib, the second generation BTK inhibitor, was approved for treatment of adult patients with CLL. And in 2020, a combination therapy involving ibrutinib and rituximab, which is a CD19 antibody, for the first-line treatment of CLL. You can also see the second-generation inhibitor zanubrutinib, which is currently in clinical trials in phase 3 as of 2017 and is currently on track to be FDA approved very soon. The non-covalent BTK inhibitors came fairly late to the clinical trials. However, pirtobrutinib is likely to be approved really soon as well. So here we are looking at some preclinical data on one of the non-covalent inhibitors, nemtabrutinib. So this is a publication from Dr. Jennifer Boyack from Ohio State University in 2018. So what they've done here is cells from a ibrutinib-treated patient that acquired C481 mutation at progression. So these cells were treated in vitro with nemtabrutinib and ibrutinib for 1 hour, and then the effect on the BCR signaling was looked at by looking at the phosphorylation of phospho-BTK. And as you can see, pre-relapse, prior to getting the C481S mutation, both drugs effectively inhibits phosphorylation of BTK, which is the blood synthase in the mouse, somewhere in the middle, BBTK 2 to 3. And then at post-relapse, on acquiring the C481S mutation, ibrutinib loses its efficacy, whereas nemtabrutinib continues to inhibit phospho-BTK. And on the right is a plot looking at the cell killing. So here, primary CLL cells were isolated from ibrutinib-resistant patients who all had the C481S mutations, and they were treated with nemtabrutinib and ibrutinib. And the effect on cytotoxicity was measured. And as you can see, there was a significant cytotoxicity compared to the control with nemtabrutinib, which is also known as RKL531. But this wasn't achieved with ibrutinib. So this is some preclinical data from our lab on pirtobrutinib. So what we're looking at here is cells from a BTK wild patient were treated in vitro with pirto and ibrutinib, and the effect of these drugs on the BCR signaling pathway, assayed by looking at the phosphorylation of proteins in this pathway, and cell death were assessed in vitro. So in the first plot and the Western blot, you can see that both drugs quite effectively inhibit phospho-BTK at all concentrations. And in the middle one shows the dose curve of inhibition of phospho-BTK, and both drugs inhibited phospho-BTK with comparable IC50s. And in the right, you can see the cell killing compared to the control, pirtobrutinib and acala and ibrutinib significantly inhibited, significantly caused cell death. So then we also looked at the effect of pirtobrutinib on a C481S in vitro model. So here, these are HEC293 cells expressing both either BTK wild type or BTK C481S, and we treated them in vitro with a range of drug ranges, pirtobrutinib and ibrutinib using a varying drug doses. And you can see in wild type, both drugs effectively inhibited phospho-BTK and phospho-PLC-gamma2. However, in the cells expressing C4H1S, pirtobrutinib continued to inhibit phospho-BTK and PLC-gamma2, whereas ibrutinib fails to have this activity. And this can be also seen more clearly in the plots on the right, where in HEC293 BTK wild type, both drugs inhibited phospho-BTK with similar IC50s, whereas in the HEC293, pirtobrutinib inhibited at 16 nanomolar, whereas ibrutinib failed to inhibit phospho-BTK. So these are CLL patient cells with the C4H1S mutation, again treated in vitro with pirtobrutinib and the two covalent inhibitors, ibrutinib and acala. And on the right, you can see is densitometry plots for phospho-BTK, and there is a significant inhibition of phospho-BTK with pirtobrutinib as 0.6 micromolar, whereas we did not observe significant inhibition with ibrutinib or acala. So here we're looking at the cytokine secretion in vitro in the supinatants after C4H1S expressed in CLL cells were treated in vitro with ibrutinib, pirtobrutinib, and acala. And similar to the beta signaling pathway that we saw earlier, we saw a significant reduction in the secretion of both of these cytokines with pirtobrutinib, whereas we did not observe that with ibrutinib or acala. So pirtobrutinib has shown promising efficacy for patients in trial. It was considered safe and active in multiple B-cell malignancies. The overall response rate in patients with CLL was 62%, including patients with C4H1S mutations at 71% and without BTK wild type at 66%. And on the right, we have another publication that recently came out, which looked at the effect on the phosphorylation of BTK in these patients who are currently on trial on pirtobrutinib. So they looked at from cycle 1, day 1, which is baseline, up to cycle 41, which is after 3 months of therapy, and there was a reduction in phospho-BTK in these patients. And we also have this other publication that came out in February, which now reports resistant mechanisms to pirtobrutinib. So they report 9 out of 55 patients had disease progression while on pirtobrutinib. And genomic analysis on the peripheral blood samples from these patients before treatment and at the time of disease progression revealed that 7 out of the 9 patients had 1 or more new mutations in BTK at the time of progression. And as you can see on the graph below, on the plot below, whereas resistant mechanisms to covalent mutations is often characterized by mutations at the C4H1 site, these new mutations were in the other regions of BTK. And some of these mutations also conferred resistance to some BTK covalent inhibitors. Another interesting thing about these mutations is that some of these lead to a kinase dead protein, as you can see in the plot on the right. They fail to have phosphorylation at the BTK Y223. However, despite this protein being kinase dead, the BCR signaling pathway appears to be intact with increase in downstream signaling molecules such as phospho-AKT and phospho-ERK. So more work needs to be done to elucidate the resistant mechanisms to this class of inhibitors to understand this further. But this will help revise the therapeutic options for these patients who's developed these mutations. So thank you for listening.
Thank you so much, Aishath. Now we will pass the presentation over to Veronique Barron.
Hi, good morning, everyone. It's a pleasure to be here from San Diego and speak after Aishath's fantastic seminar. In my presentation, I will touch only briefly on the clinical relevance of kinases, just to expand a little bit from BTK. Then I will introduce the audience to BPS Bioscience, and I will spend about 10 minutes describing some of our kinase-related tools, such as proteins, assay kits, a knockout lentivirus library, and reporter systems. So for viewers who are a little less familiar with kinases, I have illustrated a few basics. As you know, these enzymes are classified depending on which amino acid they phosphorylate: tyrosine, serine threonine, or both tyrosine and serine threonine for the so-called dual kinases. The receptor kinases are localized at the plasma membrane, and the non-receptor kinases are in the cytosol. I put separately the cyclin-dependent kinases that are a whole family in themselves. Phosphorylation is the most frequent post-translational modification of proteins, and it regulates protein function. It does so by altering the conformation of a protein, its localization, enzymatic activity, interaction with partners, and so forth. So kinases trigger signaling cascades that ultimately regulate many biological processes such as metabolism, cell proliferation, cell cycle progression, survival, death, and differentiation. Because they are involved in so many biological processes, they're also involved in many pathologies. So in cardiovascular and degenerative disease, for example, you'll find the kinase LRRK2 involved in neuron health, kinases that are involved in ER stress. They're involved in diseases of the immune system because they are part of the T-cell checkpoint regulation. They contribute to immune suppression. For example, the TAM kinases. They contribute to inflammation, as with CSF1, and they contribute to autoimmune disease. They are very much involved in cancer. So all the CDKs contribute to cell cycle progression. There are a lot of oncogenic kinases for which the mutation, amplification, or aberrant activation will directly promote tumor formation. So I indicated here a few of these oncogenic kinases, like the EGF receptor, KIT, HER2, and so forth. And they're transcriptional regulators. So at the intersection of the immune system in cancer, you're going to find a few kinases that can be involved either in autoimmune disease or in blood tumors. And then at the intersection of the cardiovascular system and cancer, you'll find kinases that are involved in angiogenesis, such as the EGF receptor and the PDGF receptor. So here are a few FDA-approved kinase inhibitors in oncology. This is a subset of these inhibitors as they target tyrosine kinases and only those that are involved in oncology. So for example, you're not seeing here the JAK inhibitors that have been approved for immune diseases. The first tyrosine kinase that was ever approved was imatinib, which is also known as Gleevec. That was initially used to target blood cancers with a BCR-ABL fusion and was later expanded to target other ABL-linked tumors, such as a gastrointestinal stromal tumor. It was followed shortly after in 2002 by the first generation EGF receptor inhibitor, which is gefitinib. And all these early successes have opened the door to further development of small molecule-based targeted therapies because of their high efficacy and because of their large market shares. So you're seeing the number of small molecules that have been approved increase over time. And to the right, I also added the 2021 approval for tivozanib and trilaciclib. Even though CDKs are not tyrosine kinases, but I wanted to showcase this because they are the first CDK inhibitor approvals. So now, not all drugs against kinases need to be small molecule inhibitors. So for receptor type kinases, for example, that have an extracellular domain outside the cell, they can also be antagonized using extracellular antibodies. So what I'm showing on this slide on the right are 3 FDA-approved monoclonal antibodies against the EGF receptor. And then I also indicated the 3 generations of small molecule inhibitors, with the light blue being the first generation and the dark blue being the 3rd generation. And this is to keep in mind that once a drug against one kinase is in the clinic and is showing efficacy, there will be additional drugs that will be designed, just because as we learn more about the side effects of the drug, or we learn more about mechanism of resistance, we need better drugs with better affinity, or drugs that can recognize a mutant that pops up during resistance, for example. So if you're working on kinases, what are the tools available to accelerate your research projects? So to study kinases in vitro, you may want to look at the structure of some of the protein domains, for example, the kinase domain. You may want to analyze binding partners, such as drugs, antibodies. You may want to design a novel assay. And very often scientists will use purified proteins, either in white type form or in a mutant form. They may want to tag these proteins. In addition, they will want to measure the kinase activity, and they may use a kinase assay kit. Now, to study a kinase in a cellular context, you can overexpress this kinase in a particular set type of interest. A target validation study may also use CRISPR-mediated knockout, or may compare the wild type and the mutant forms. The biological function, and especially the activation of relevant selecting pathways, can be conducted using cell lines or using reporter systems. So now let me introduce my company, BPS Bioscience. We are a scientist-founded, scientist-driven company. Our mission is to advance new scientific discoveries that lead to therapies by creating innovative solutions for research. The company was established in 2005 by Henri Zhu, Dr. Henri Zhu, PhD. We're innovating drug discovery in various spaces, such as immunotherapy, epigenetics, SARS-CoV-2, and cell signaling. We are industry leader in recombinant proteins and hard-to-purify complexes. We're also a leading producer of recombinant cell lines and cell-based assays. We're dedicated to delivering novel assay kits, cell systems, and virus-based tools for research. Our core technologies are summarized here. We can provide custom proteins and conjugates. We specialize in high purity, high yield expression of recombinant proteins. Formulation, activity testing, labeling can be customized to research needs. Cell line development. We provide stable cell lines that express validated surface proteins and reporter cell lines to probe cell pathways. Our expertise with CRISPR-Cas9 also enables the manipulation of protein expression in cells, either by knock-in or by knockout. Virus engineering, we provide high-titer lentiviruses, VSVs, and AAVs, adeno-associated viruses, that can be used for viral infectivity assays, stable cell line generation, CRISPR knockout, and much more. We also provide screening and profiling services. We save you time by evaluating lead compounds with our biochemical and cell-based screening services. And coming soon are our iPSC-derived cells. So we'll provide differentiated cardiomyocytes and neural cells derived from induced pluripotent stem cells. So for proteins, they are used to study the structure, the effect of a mutation, the binding of an antibody, to design assays. For receptor tyrosine kinases, most of the time you will want only the kinase domain. Of course, we cannot purify the full protein because it would lack the plasma membrane, so it wouldn't be folded correctly, but will produce either the extracellular domain or the kinase domain. And again, expression can be done in bacteria, insect, mammalian cells, available in wild type or mutant forms. Multiple tags are available. They are affinity purified. Formulation can be customized, and we always will test the activity as part of the quality control. Kinase assay kits will allow the measure of the activity of the kinase, drug kinetics, and assay 50s. They're very simple assays, really. You add the kinase and your drug, pre-incubate them, add the substrate and ATP, and then add the kinase glow and measure the signals. So their assay kits are non-radioactive. They're provided with a detailed protocol. The kinase, the substrate, the ATP, and the buffer are all included. They're accurate, robust, and specific. And I put to the right two graphs that were opened with two highly related kinases, ROC1 and ROC2, studying an inhibitor and measuring the EC50 of this inhibitor. We also have fluorescence polarization assay kits. So these assay kits have been specifically designed to probe the GH2 pseudokinase domain of the JAK family of proteins. So the JAK family of kinases that include JAK1, 2, 3, and TYK2 play very important roles in blood cancer and in immune diseases. GH1 is the kinase domain, and GH2 is the pseudokinase domain. And at the bottom, you see an illustration of the linear structure of the protein with the different domains. And then you see a little illustration of a proposed conformation and how these domains interact with each other. And what you'll see is that the GH1 domain and the GH2 domain are very closely interact with each other very closely. The pseudokinase domain have very weak kinase activity. And so you can't really measure its activity using a kinase assay. GH2 domains, however, play a role in the function of the kinase, and both the GH1 and the GH2 domain are required for signaling. So the GH2 domain does bind ATP, which accounts for the pathogenicity that is associated with mutations in the GH2 domain. And there are many mutations in this domain that have been found in leukemia, in various forms of leukemia. So the ATP binding pocket of GH2 is accessible to small molecules, and it is therefore a very attractive therapeutic target, especially because it is expected that inhibitors against GH2 may be somewhat less toxic than existing inhibitors against GH1 domains. So the fluorescence polarization assay kits bypass the need for a kinase assay, since there is no kinase activity for the GH2 domain. They consist of a purified GH2 domain, for example, for JAK1 or JAK2 or TYK2. You have a small probe that is labeled with a fluorophore. So when the probe is free, it has low fluorescence polarization. Upon interaction with GH2, it forms a much larger complex, and the fluorescence polarization is now high. So the formation of the complex leads to a higher signal of fluorescence polarization. In the presence of the inhibitor, the small molecule blocks access of the probe, so the probe remains free and fluorescence polarization remains low. So this is indicated here. This is on the right, the binding of the GH2 probe 1 to the JAK2 GH2 domain. And as you can see, as you increase the amount of GH2, you also increase the signal because more and more of the probe is going to be associated with GH2. Now, if you were to add an inhibitor, you would be able to measure the decrease in signal that is induced by the inhibitor, and you would be able to measure the Assay 50 for your inhibitor. So let me tell you a little bit about knocking out the kinases. So this is to help with the validation studies. We have generated a knockout kinase library, which is based on CRISPR-Cas9 technology. This is a library of lentiviruses. They're off-the-shelf viral particles, and they're all in one system, which means that the viral particle, the lentivirus, will transduce the Cas9, the single guide RNAs that are needed for the knockout, and they also contain a pyromycin selection gene. So you can use them with any cell of interest, including non-divided cells, regardless of whether your cell contains Cas9. So you don't need to use a Cas9 expressing cells to use this lentivirus library. Now, every vial of the library targets one gene, but every vial also contains 5 different single guide RNAs, and this is to make sure that we get the best knockout that is possible. So each vial contains 5 sgRNAs and targets one gene, and then the full library, each one is individually constructed, sequence verified, and purified. The full library covers 619 human kinases and pseudokinases, 5 sgRNAs each. It also contains 150 control sgRNAs, and they can be used in a biosafety level 2. So the applications are broad. They go from viability screening. You may want to identify kinases that are involved in survival. You may want to do a synthetic lethality screening, drug resistance study, and so forth. It is possible to identify which kinase activate a transcription factor by using a reporter cell line for transducing the library, for example. So that brings me to our reporter systems. These are used to test the activation or the inhibition of a particular cellular pathway using a high-throughput screening. We have over 20 available cell lines, 70 available reporter systems. It is based on the activation of a particular transcription factor of interest. This transcription factor, TF, will bind to the response element, and this will induce the transcription of the reporter that can be either luciferase or can be fluorescent protein like EGFP. Once the reporter protein is expressed, you can measure the expression either through immunofluorescence studies or through measure of luciferase activity and luminescence assays. So in the case of EGFP or RFP, which is a red protein, you can do flow cytometry, microscopy. And with luciferase, which is a luminescence system, you can read the signal using a luminometer. This is a very robust, quantitative, and reproducible measure of signal activation, and can be very useful to see if your kinase is active and which cellular pathways they're working with. So we have a few promoter-specific reporter cells that are off the shelf, ready to go to measure the activation of transcription factors such as AP1, CRIC-REB, CSS, and so forth. And they exist in various cell types. We can also make the reporter cells that you're interested in if you don't find it on our catalog. So on the right, you see, for example, the measure of NF-kappa B activation by agonist TNF-alpha on top. And then at the bottom graph is a measure of the inhibition of NF-kappa B by a specific inhibitor. If you would prefer to make your own reporter cells, you could use lentiviruses. These will allow for the robust, quantitative, and reproducible measurement of transcription factor activity. Again, we have over 70 reporter systems available, and they are useful to study transcription factor activity. And I might point out here that it could be for short-term studies. You can do a transient transfection and measure the activity of your transcription factor in response to any signal or with an inhibitor. But it can also be used to generate a stable cell line using the antibiotic selection that we have in the lentiviruses. Determination of IC50 or EC50 is possible, and you can screen inhibitors in cell-based assays, which is very important to look at a cell-relevant response. Finally, our CRO services will help scientists evaluate lead compounds with biochemical and cell-based screenings. We have a unique panel of over 500 assays. Our 3,000 square foot facility is fully equipped with state-of-the-art instrumentation, and we also have biolayer interferometry for protein interactions. With all screening and profiling services, we provide a comprehensive report that contains raw data and analyzed data, all the graphs and detailed protocols. And since we produce our proteins, our enzymes and cell lines in-house for this assay, customers can trust that our quality and consistency are of the highest level. And we are your partner in drug discovery. Thank you. We will take any questions now.
Thank you, Aishath and Vernick, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, several tyrosine kinases are implicated in BCR signaling pathway. Diagram in introduction indicated lin and Syk tyrosine kinases upstream of BTK. Are there inhibitors being developed against these kinases as well? And do you know if such inhibitors show any kind of efficacy, at least in preclinical data?
Clinical data, lin and Syk inhibitors have shown efficacy. As to whether any of these are in clinical trials at the moment, I have to double check that. But having said that, a couple of the non-covalent inhibitors that I showed in my presentation today, nemtabrutinib also inhibits Syk, so that hits Syk and BTK. And another one, CG806, which is in preclinical development, also is a dual BTK-Syk inhibitor. So yes, targeting all the molecules that you mentioned is definitely beneficial to getting a larger inhibition on this sectoring pathway.
Great, thank you for your response. Question 2. Is mutation of cysteine 481 the only genetic modification seen in ibrutinib-resistant patients? Are there other mutations seen in BTK? And if yes, have you tried pirtobrutinib on some of these other mutants?
So the C481 site appears to be the predominant one, and other than cysteine, there are other substitutions that happen at this site, and it has been shown in preclinical data and in clinic that pirtobrutinib is active against those. There are, however, a very small percentage of patients that shown a couple of other sites, and now that we have seen resistant mechanisms on non-covalent inhibitor pirtobrutinib in trial, we now know that pirtobrutinib is also resistant to those mutations. So there are some mutations that are common to both.
Okay, thank you. It looks like we have time for one more question. And this question is for you, Veronique. You showed that BPS has a knockout kinase lentivirus library. Can I order a single lentivirus if I'm interested in only one kinase?
Oh, yes, actually, absolutely. I skipped over this maybe a little fast. So the library is made of single vials, and it's possible to order any single kinase that you want to target. We also offer panels, which means that you can, if you're studying, say, the JAK kinase family of proteins and you want all four of them, you may just order these four. So we have either individual kinases, panels, custom panels, and the full library.
Okay, thank you. Okay, it looks like that's all the questions we have for today. Thank you, Aishath and Veronique. Do you guys have any final questions or comments for your audience? Okay. All right.
Well, I would like to say thank you to Aisha. That was a very interesting presentation. I'm really, really happy that you were here to present to us today. So thank you very much.
Thank you for the opportunity. I'm glad. And as a final comment, I think I would like to say that BTK inhibitors, with its increasing proven efficacy in CLL, is become a mainstay in CLL treatment. So.
Great. All righty. Well, thank you again, Aishath, and also to Veronique for your time today and your important research. We would also like to thank LabRoots and our sponsor, BPS Bioscience, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye.